Yeah, so as Mark said, I work on a bunch of different things. I'd say I'm an imaging scientist who runs a computational imaging lab at this point. And, we have some projects across modalities. The common thread is that, there's usually an inverse problem of some kind to solve, and ideally some interesting physics to model. I'm really most interested in getting the right forward model and then finding ways to invert it. And so we've worked in spectral computed tomography, different ways of acquiring and reconstructing multi-channel images. These are not patient images, but actually images of a frozen turkey that we acquired. That's why they have, you can see there's 3 different vertebrae that you can see because of how a frozen turkey has its neck folded up inside the body cavity. At a different resolution scale, we've done a lot of work at the advanced photon source with very, very high resolution CT. So CT with 1 micron voxels of metal stain model organisms like zebrafish. And so this gives essentially cellular resolution, nearly, nearly histology level resolution. But across an intact 3-dimensional organism with isotropic resolution. And so this can be useful for phenotyping. We've started using this to do validation of diffusion MRI and tractography. So mouse brains, we can take a sort of ex vivo mouse brain and do sort of super high quality ex vivo MRI on it, and then stain this brain with heavy metals and do synchrotron imaging at micron resolution and actually be able to sort of detect, the axons that are not individual axons, but sort of, bundles, and, begin to use that to validate the findings of the DW MRI. Yeah, and these same samples after X-ray, they've been stained with heavy metals, the same heavy metals you use in electron microscopy. And so you can take sort of a small region out of it and sort of zoom in and do sort of nanometer level resolution. And this pipeline is not smooth yet or easy to do, but ultimately the hope might be to sort of look for interesting areas maybe where DW-MRI is having trouble capturing something, and then really look at, or maybe where it's capturing it well, and then really look at the ultrastructure of that very voxel using EM as a way of understanding what's going on. We've had some nice projects with Lingjiang Meng from Urbana-Champaign, who I think many of you know, on X-ray fluorescence tomography. But what I want to talk to you about today is some of the work we've done in computational microscopy that sort of fuses this idea of multi-view deconvolution with this idea of orientation imaging. And I won't dwell on this slide because this is what my talk is going to be about. And this work started a few years ago when the University of Chicago affiliated, in some ways, sort of took over the Marine Biological Laboratory in Woods Hole, which is a very, famous and storied center for biological research and especially for education. In some ways, it's the National Biology Lab of the United States. It's just not actually a federally funded national lab. It's a private nonprofit lab that has a very long history in microscopy and education, and they were having a sort of a financial emergency. And so now they're basically part of the University of Chicago. And it's a place that attracts a lot of people from around the country who come in the summer at other times of year, as well as there are some year-round investigators like Rudolf Oldenburg, who's sort of a leader in polarization microscopy. Hari Shroff is a leading microscope developer. He's an intramural at NIBIB at NIH, and he's been coming there for many, many years. And most of the actual hard work on this project were done by my student, Alan Chandler, who's sort of now a master of image science and has just started a postdoc at UBC, and then Min Guo from Hari's lab, who did the hardware side of things. And so the story I want to tell you is one about fluorescence and anisotropy. So this is about microscopic images using conventional fluorophores like green fluorescent protein or other similar dyes, and trying to extract some additional information from them. And so I'll start here. If you look at a conventional microscopy image like this, if you could keep zooming in, if you could really look at the fluorophores themselves, at sort of the highest possible resolution, what would you see? Would you see sort of an isotropic emitter, sort of a light bulb emitting light, equally in all directions? Like we think about in nuclear medicine, an isotropic emitter. No, actually, it turns out, and some of you who are in optics may well know this, that fluorescent molecules are actually all dipoles. They're sort of tiny little optical antennae, and they emit light, the fluorescence with an anisotropic pattern and with specific polarization. Okay. We can usually ignore this because we're looking at ensembles of molecules that are all randomly oriented, or looking at individual molecules that are tumbling around rapidly and averaging out this effect. But there are ways to exploit this effect, okay? And so the goal, what we're aiming for in the work I'm describing here, is to sort of not see sort of little spherical balls of fluorescence emission, but to actually be sensitive to the orientation of the fluorophores, and to use that to report the orientation of structures that might be below the resolution limit of the microscope. Okay, so a little bit about polarized fluorescence. We won't get too much into the details. The usual modeling that we do for these random ensembles or rapidly tumbling molecules sort of pretends that they're a monopole, like an isotropic emitter. But in fact, as I just said, any real, single sort of fixed, rigid dipole, fluorescent molecule is going to be typically a dipole or even higher order emitter. And so it's going to emit strongly, at 90 degrees to the dipole axis, not at all parallel to the dipole axis. And it will emit polarized light, and it will also be excited differently depending on the the polarization of the excitation light. Okay. So how can we exploit this to image the orientation of molecules? Okay. Thinking about it from the standpoint of excitation that I just mentioned, so if we have a dipole with a particular orientation, you can think of this dipole as sort of a vector. Actually, it's probably better to think of it as a double arrow vector. We can't tell up from down. There's a symmetry if you flip them 180, but it has an orientation. It's pointing along some line. If you excite a dipole with polarized light and the polarization vector is parallel to the dipole, you'll get a very strong emission, a very strong signal. If you excite the dipole with polarized light that is perpendicular to the dipole, if it's truly perpendicular, you'll get no emission. Okay, the excitation efficiency is given as the dot product of these two vectors, and so that would be 0. So it'll be no or weak emission. Okay, and this was shown in a very sort of compelling way, about 20 years ago by Shinya Inoue, who was at the Marine Biological Laboratory. It's one of the sort of founders of the field of polarization microscopy, and he just took a bunch of green fluorescent GFP crystals and sort of threw them down on a slide, and then excited with light in this left-hand panel. The light is horizontally polarized, and so you can see that the horizontal crystals are very, very bright, and the vertical ones, quite dim. And then over here in the right-hand panel, the polarization is vertical, and you can see the vertical ones are lighting up. Okay, so this is a way, if you couldn't see, these are big crystals, we can actually see how they're oriented at the scale of the microscope. But in principle, this would allow you to be sensitive to even sub-resolution orientation, and that's what we're trying to exploit here. Okay. Okay, so how are we going to use this? We're not interested in mapping the orientation of sort of naked fluorophores of loose solutions of GFP. they need to be linked rigidly or bound in some predictable orientation to macromolecules of interest. So that's what people do. And we haven't, I should make clear, we didn't invent this technique of fluorescence anisotropy. We've just tried to do it in a sort of more three-dimensional way than anyone has before. And so what people have done is you take a protein of interest. So here's a septin, and they link to it a GFP with a very rigid linker. And the rigidity is important. You don't want the GFP to be flopping around and tumbling around, because then you're going to average out all these polarization effects and you're not going to see it. You'd like to know that if you detect the orientation of the GFP, here being kind of vertical, it's telling you something. It's telling you that this septin is kind of, oriented orthogonally. Okay, and so Tim Mitchison at Harvard had a very nice paper in Nature in 2006, where they made this septin rigidly linked to a GFP and used sort of a basic single view polarized fluorescence microscope, varied the polarization of the illumination, and they were able to show, during cell division in yeast, it was known that you get kind of this hourglass shaped structure of septin. And they were able to show that there's basically two different orientations of septin, that these rings up top have the septins oriented in one way, and the sheet connecting the rings have the septin connected in the other way. And this is not something you can see these individual septins with the resolution of any microscope. You couldn't detect this structure except through this fluorescence anisotropy measurement. So that was a nice example. Okay, so what are the current approaches to orientation imaging? How do you actually do this? So I think prior to our work, everyone had been using sort of a somewhat conventional microscope, either wide field or confocal, looking from a single view direction at the microscope. So looking, say, down the optical axis. And what you're trying to detect essentially is, if you have, within a resolution element, some ensemble of oriented dipoles, you can picture this as, I'm showing kind of a cone of vectors. You can think of them as, being contained within this cone. I mean, this cone has some spread, it has some orientation, some angle theta with respect to the z-axis, some angle phi. And the goal of these imagings are to try to image this. Okay, and usually what you would do is you'd be sort of looking down the optical axis, and you would excite, say, with 4 different polarization settings, 3 or 4 different polarization settings, and sort of doing some arithmetic allows you to infer this angle phi fairly well, and kind of infer. Basically what it allows you to do is to learn something about the length of the vector that gets projected down into the XY plane. And so, the length and its phi. Okay, so the phi you can pick up reasonably well. The length is some kind of complicated combination of what is the polar angle, what is theta, and what is the spread around the cone. Okay. And so, pardon me. So there are some limitations to that, problem of looking from a single point of view at the projection of a group of vectors into a plane. Okay, so we can't sort of separate the effects of this polar angle theta from kind of the spread of the cone very well. We would typically estimate kind of a mean orientation. And so, in a more complex system, if we had crossed dipoles, the best we could hope for from the single view system would be to estimate the, the mean direction. And then we wouldn't be able to distinguish dipoles that are above or below the focal plane. They would both project into the focal plane in the same way. And so what we started thinking about, Hari Shroff at NIH, for other reasons, had built this dual view light sheet microscope. Okay, so this light sheet microscope has 2 different objective lenses, A and B. They're, in its simplest version, they are basically the 2 copies of the same lens, and you can illuminate through either lens and detect through either lens. And he had developed this mainly to address the problem of anisotropic spatial resolution in light sheet microscopy. The fact that you get good XY resolution for any plane that you're illuminating, but the thickness of the slice always ends up being 2 or 3 times worse than what you can do in XY. And so he made a microscope that could image 2 different stacks of the object, one of which has poor resolution along one axis, the other one has poor resolution along a different axis, and he was interested in computationally fusing those 2 volumes to get isotropic resolution. Since this already existed, our thought was, if we add polarizers to this, we would be able to sort of look at these dipole vectors from 2 different angles, and this might begin to resolve some of the ambiguities and limitations that I just described to you. Okay, and so then, the basic idea then is to, we're going to, illuminate from one side with 4 different polarizer settings and acquire a stack, measured in the other objective, and then switch, illuminate from side B, 4 different settings and acquire through stack A. Okay, and so the kind of data we get, this is the same slice once you've kind of registered those 2 volumes and extracted the same, rotated them onto each other and extracted the same slice. This is the same slice, from a volume seen from, camera A, from camera B, and this is with the 4 different polarizer settings. There's not much to see here other than the fact that, as we change the polarizer setting, there are some changes in local changes in intensity in different parts of the image because there are oriented structures here. This is we've labeled this structure with a dye that should bind to the actin filaments in a sort of predictable way, kind of tangent to the actin filaments. Okay, and so my interest and that of my student was, okay, how do we process this data? How do we reconstruct this data and get orientation information from this in the 3D volume? The kind of simple arithmetic that had been done in all previous microscopes didn't really work here. There was no way to, you could kind of do it on one stack, do it on the other stack. There was no way really to fuse them in any meaningful way. And so this ended up being a really fun intellectual adventure. This student, Talon Chandler, has really created a whole new sort of theory for the field of spatio-angular imaging, rooted in image science and in the kinds of object space to image to data space mappings described in the Foundations of Image Science book by Barrett and Meyer. And so the first was to think about what is it that we're actually trying to reconstruct? What are these images that we're trying to make? And we realized our goal was basically at every location in space to be able to reconstruct the distribution of dipole orientations. Okay, so we need to reconstruct an orientation distribution function at every spatial position. So you can think of this as a sort of a probability distribution function on the sphere, right? Probability distribution function of directions. Yeah, so an ODF that looks like this means that we've got lots of vectors, pointing vertically. Okay, and all the ODFs I'm going to show you are going to have, up, down. We're going to have a 180-degree symmetry because of what I said before. If you flip these vectors, if you flip a dipole, it gives you the same. It responds in exactly the same way. So we're never going to be able to tell a 180-degree flip. Okay, so this is what we're trying to reconstruct. Is we'd like to know at every point in space, what is the distribution of these dipoles. We'd like to know. So in one limit, if they're isotropically distributed, our ODF would just be a sphere. We'd have equal probability of finding a vector pointing anywhere. If we have a concentrated ensemble, if we have a fiber, a single actin fiber coming through, we would expect an ODF that looks like the middle panel. And if we had 2 crossing fibers, 2 crossing ensembles of oriented dipoles, we might expect something like on the right. And okay, so mathematically, what we're trying to reconstruct is some function f that's a function of 3 spatial coordinates, x, y, z, where are we in the image volume, and then 2 angular coordinates. At that position, what is this orientation distribution function? Okay, so this f of r s hat is what we're trying to reconstruct. Okay, so what do we measure? Oh, sorry. The other thing that we need to model in our forward model and deal with is this issue of anisotropic spatial resolution in a light sheet microscope. Frankly, in almost any microscope is going to have poor axial resolution, right? Can have good transverse resolution and poor resolution along the axial direction. And a light sheet is no different. I mean, the axial resolution is set primarily by the thickness of the sheet that you bring in. But for a variety of reasons, that can't be made quite as good as the XY resolution typically. And so the view A, if you're just looking at a conventional fluorescent sample, none of these angular effects I've just been talking about. If you just look at a sagittal slice, extracted from this plane, from this stack, it's going to look blurred in the Z direction. If we pick up the view B stack and pick out, the corresponding slice, this is exactly the same slice from the other stack, it's going to be blurred in the X direction. Okay. Remember, I said the purpose of this dual view microscope, and the reason it was invented in the first place was not to solve this polarized fluorescence problem, but was to solve this problem: the fact that your images are always going to be blurred, your 3D images are always going to be blurred in the 3rd dimension. And so, Hari's solution was to, computationally fuse using Richardson-Lucy, these 2 volumes to get, much more isotropic resolution. Okay, but we still have to model this effect. This effect doesn't go away in our data. Okay, so the forward model that we were working with is the following. Okay, so here's this spatio-angular density that we're trying to reconstruct. This is what we're after. Okay, over here on the left is our measurement. Okay, so our measurements, for any given illumination of the light sheet, our measurement is a 2-dimensional function, right? It's sort of a frame captured by the camera. So RD is a 2-dimensional vector on the camera. I guess in the limit of a stack, we can think of it as 3D. And then we've got this discrete index I. This is because we can change the polarizer to 3 or 4 discrete settings. And this I can also index whether we're looking from view A or from view B, from camera A or camera B. Okay, so this is what we measure. This is what we want to reconstruct. And the physics and the image science really comes in this kernel, this spatio-angular point spread function. And so this is basically telling us, if we put, a point source, a delta function at R0 that has a particular orientation S0, is also a sort of a delta function on the sphere. This H of I is what does our data look like? Okay, what does the image stack for polarization setting I and view A or B look like for that? And so our goal was then to understand what are the properties of this equation and how can we invert it? What are the properties of this kernel? Okay, so stepping back, this is not too complicated. It's a linear equation. It looks like a pretty typical, continuous to continuous mapping. We've got this extra sort of angular integration. Let's step back to sort of a simpler and more familiar example. If we throw away, if we consider just a conventional spatial microscope that is where these angular issues are ignored or can be ignored because of isotropic distributions of emitters, and if the microscope is shift invariant, then that kernel H, if you focus here on the middle, that kernel H becomes H of RD minus R0, becomes a convolution essentially. And we know that one of the best ways to reason about these kinds of equations is to choose a new basis, a convenient basis that makes it easy to think about what is going on, and also easy to invert equations. In this case, for a shift invariant system, we know that the most convenient basis is the basis of complex exponentials. It's to take a Fourier transform and move into frequency space, and we end up down here where the convolution becomes a multiplication. Okay, and so our object function f becomes capital F, and our point spread function becomes a transfer function. And this transfer function point of view is very useful because we know that there is, that often these transfer functions will have some finite bandwidth. There's going to be some frequency at which they go to 0 and remain at 0. And that tells us something about, what is the highest resolution feature we can hope to recover in our reconstruction. It tells us something about the null space of the imaging system, right? What elements of the input space of the object are invisible to the imaging system, get mapped to 0, okay? So, this should be, I think, familiar to most of you. What we're going to do with our problem is something similar. We just need basically 2 changes of basis, okay? The first assumption we're going to make, which I think is quite reasonable, is down here at the bottom, that our microscope also is shift invariant, okay? And so that we can express it as just the difference between RD and R naught. Okay, and so with our microscope being shift invariant, that again brings up the complex exponentials as a natural basis to move into for the spatial part of the problem. For the angular part of the problem, the right basis to go into to make things simpler is to go into the basis of spherical harmonics, which is a basis for functions on the sphere. And some people refer to this as, these are the YLM of S naught, and these expansion coefficients here, which are indexed by 2 discrete indices, L and M. Some people refer to this as the spherical Fourier transform of a function, okay? And so here's a zoom of these spherical harmonics if you're not familiar with them. They're indexed by 2 indices, L equals 0, or, L goes up, and you can see that the features get more complicated, more high resolution as you go up in L. And then for any given L, as you go up in L, there are increasing numbers of M basis functions, and these give you sort of some more orientation information. And so it turns out that, for this problem, and again, we've got object space on the left, data space on the right, focus again, in the middle here, we've got this convolution-like feature in the spatial part. We still have the angular dependence. An important point to make is that this kernel is not separable. We can't factor it into, okay, spatial stuff happens over here. Angular stuff is governed by a totally different function, and govern them separately. They stay together. But if we do take both of these basis transformations, we do end up with sort of a generalized transfer function that depends on spatial frequencies and on the polarizer orientation, okay? And it again has sort of the form of a product in the spatial coordinate. We just also have this extra sum over the discrete indices of the spherical harmonics, okay? And so, the interesting question we want to, one key question we wanted to look at is, what can we say about the null space of this transfer function? Does this tell us anything interesting about, for example, which spherical harmonics are actually passed by the microscope, okay? And it turns out it does very powerfully so. And I think this was one of Talon's most important results. I don't think this was widely appreciated in this field, that you can really carefully characterize the null space of a wide variety of different microscopes, sort of the angular null space or angular band limit of microscopes. And so, yeah, you can sort of clearly show that microscopes only pass a finite number of these spherical harmonics. So a single view microscope with no polarizers can only pass the spherical harmonics shown here in square, surrounded by squares. One thing I should point out, we're never going to pass, or the odd spherical harmonics, the L equals 1, L equals 3, L equals 5, those are always irrelevant in our problem because of the mirror symmetry I've mentioned a couple of times, because we don't need them to represent a distribution function that has mirror symmetry. Okay, so those are always irrelevant. It's just the even L equals 0, L equals 2, L equals 4 bands. Okay. So a single view microscope with no polarizers. So this isn't even the kind that people were using before to do the simple stuff I showed you. Those had polarizers. This is just a conventional microscope. It does have a little bit of, a tiny bit of orientation information gets passed. It is still sensitive to, if it had a really, really small numerical aperture, you could potentially have, a dipole pointing straight up at it. And if you excited it in the right way, you would never detect any of that information because it would all be directed away from the lens. Okay, so there is a little bit, and that would give you some information about orientation, but not much. Okay, if you add polarizers, if you pick up, 3 polarizer settings, then we do expand the angular, pass band of this microscope. We pick up these extra spherical harmonics, okay? And this is what begins to give you some azimuthal information, that phi angle we saw before, okay? That would be invisible here. You can't represent an ODF that, lacks phi symmetry, azimuthal symmetry, unless you have these spherical harmonics that correspond to m not equals 0. And so picking up these extra spherical harmonics is good. However, we still have these gaps, okay? We still have these, sort of blind spots, and that's undesirable. It means that there are certain, distributions of orientation that we would be insensitive to. And it basically means that we would not have this, that our angular resolution would depend on the angular orientation. And that's sort of the angular analogy of having your spatial resolution be different at different spatial locations in your image. And that would be undesirable. Ideally, we would like to fill in a complete band, of M values for one of these L values, okay? So we thought that just the plain old dual view microscope, just looking at something from 90 degrees would break a lot of the limitations that I described to you earlier. It turns out that it helps, it fills in one of these gaps. We pick up this guy, but there remained a blind spot that we had not anticipated. I'll show you a figure later. We were able to solve that by sort of tilting the light sheet, basically to probe, one additional direction that wasn't getting well probed. And when we do that, we're able to sort of extend the pass band, the angular pass band of this microscope, to include these complete, potentially these complete orientation bands. Okay, sorry, where'd I go? Yeah, and so, in practice, the point of all this is, the following. If this is a true object distribution, some, crossed fibers, represented by this fairly complex ODF. If you tried to reconstruct this with a single view microscope with polarizers, the best you could do, what you would reconstruct would be this one, this view here shown in the center. And we can see this is sort of completely erroneous. It has the orientation, it's kind of averaged these 2 cross directions and giving us something completely erroneous. The dual view reconstruction we can see comes much closer. It has finite angular resolution, and we expect that because we're only getting up to a certain L band of spherical harmonics, but it's at least getting the key directional features correct. Okay, so that characterizes the, the forward problem and the null space to some degree. The other nice thing, if I go back to this slide and we focus again in the middle, is the reconstruction becomes much simpler when we go into this basis, because all we have to do is, our reconstruction involves reconstructing this capital FLM of nu. And this is only going to be reconstructible, non-zero, for a very finite number of ln m. Okay, so instead of reconstructing f of r naught s hat, where s hat can take on any possible value on the surface of the sphere, as sort of a continuous function on the sphere, or if we discretize the problem, could involve, tens of thousands of sample locations on the surface of the sphere. Down here, we've simplified our problem as one of reconstructing just a finite number of spherical harmonic coefficients. And we know there's no loss of information because only a finite number of them are estimable in the first place because of the null space that we characterized. Okay, and so I won't dwell on the mathematical details of the inverse problem, but basically we take our data, we decompose it into spherical harmonics. At every spatial location, we take a Fourier transform of these, and then we solve a very small, sort of 15 by 15 SVD problem of a matrix at each spatial frequency, and then use a regularized SVD-based inverse to calculate those coefficients, and then we can come back into the spatial domain and represent ODFs. Okay, so I'm going to end by showing you sort of the kinds of images we can reconstruct, and this is still a work in progress. Sort of the killer app image is, we're still acquiring or hoping to acquire at NIH, but these were just some test images. So these were xylem cells from tobacco plants that have a helical cellulose pattern labeled with a dye that's known to bind tangent to the helix. Okay, so the dye should be oriented along the helix. And what I'm showing you here is in the left column, the bottom row is a zoom of the top row. The left column is sort of the full orientation distribution function. The center is the peak, which way is what direction is the peak direction of that ODF. And then the right is just the conventional fluorescence image you get by integrating out this angular information, right? If we just integrate over this S hat, that just tells us how many fluorophores are at that location, which is what a microscope typically measures. Okay, so you can see in the density image this helical structure, and we can generally see, and if you're interacting with these live, you can see that these ODFs are indeed generally oriented correctly along the sample. And that, you can see also in these peak directions that that's the case. Oh yeah, forgot that this was live. And yeah, these are in fixed cells that have actin labeled with phalloidin, again, a dye that should bind sort of tangent to the actin filaments. And again, we're seeing sort of similar, similarly encouraging. I mean, we do see some weirdness. These were acquired before we implemented the tilting to try to solve the sort of final blind spot. So we do see a few sort of orientation artifacts. Yeah, this is, the real goal is to do this live. These were done, this is a full volume every 20 seconds. And there's ways to try to make it even faster, but the goal would really to be able to see sort of order emerging, to see sort of the cell putting an ordered structure together and being able to detect that order, to see are these subunits of actin somehow kind of swinging into orientation before they get hooked up together, or at what stage does that happen? Yeah, this is, I've shown you this one before. This is just coming back to this point, where we got surprised once we did this careful null space analysis that our initial design, that our initial, intuition that just having the two views would be enough to close the gaps in our null space was not enough. We had to add this light sheet tilting, basically to kind of swing the laser beam a little bit, sort of to pivot it around the point at which we're trying to create the light sheet, so as to have a little, so that the polarization vectors had a little component, at least in the y coordinate, that they wouldn't have had otherwise. And that's what allowed us to fill in that sort of gap in the null space. And this we tested on, this is kind of a biological phantom. This is something called a giant unilamellar vesicle, a GUV. And it's a kind of a membrane, a bubble-like membrane. And these dyes are known to stick sort of axially, radially through the membrane. So it should look like a koosh ball, the reconstruction. And that's what we're seeing. The ODFs are generally kind of sticking out radially, and we can see that these that the peaks are generally in the right direction. The color coding is sort of indicating the general direction, and these are approximately consistent with what we would expect. Although if you look at it, oh yeah, this is a zoom. There's a little bit of, oh no, sorry, this is the one that was done properly. So this one, these generally agree with what we would like. This is the comparison that he made between, on the top, not tilting the light sheet, and on the bottom, tilting the light sheet. And, basically we, this slice on the top is sort of showing this, this kind of comb over here is indicative of this orientation blind spot. And that gets fixed with this tilting. And we recover these orientations, which should be pointing sort of straight up and straight down at the top and bottom, and not all lying horizontally like they seem over here. Okay, and so this was a nice example where sort of careful image science theory, informed a change in the hardware that we were then able to show fixed our problem. Okay, so to wrap up. I hope I've convinced you that multi-view imaging combined with, appropriate mathematical and computational modeling, it's really that combination of the hardware and the computation is what allowed these reconstructions of dipole orientations truly in 3D with time sensitivity. This was all done with excitation side polarization optics. And that's part of what slows us down. We have to acquire the volume over and over again with different polarization settings. So adding it to the emission side would speed things up. We could sort of we could sort of feed the same output light, split it through different polarizers, and get all that information at once. There are still some limitations. I mean, the angular band limit, the reason we only go so far up in the table of spherical harmonics is fundamentally limited by the physics of excitation, there's kind of a cosine factor from that dot product I mentioned earlier, and that gets squared when we excite. And, that sort of limits how high of a angular probe you can have. But 2-photon excitation could potentially double that. So that might be something to look at. And then you could also potentially look at nonlinear effects to further improve angular resolution. And yeah, I think that's it. The theory papers that begin to lay out the forward model have all been published in JOSA A in the last a couple of years, shown here. And then we're sort of acquiring more data for the applications paper. And with that, I will stop and ask if you have any questions.
Thank you, Patrick. That was really a fantastic talk. Really amazing results. Let's see if any questions from the audience.
I guess I can start perhaps. So yeah, thanks, Patrick. That was a great talk and really wonderful work. I guess my first question is whether the ODF is affected by any sort of partial volume. I mean, so I would think that if you were discretizing part of the spatial part of your function using voxels, then the size of that voxel would somehow skew the distribution, the angular distribution. But perhaps in your case, I guess you're not really explicitly doing that. You're deriving your inversion formula in a C to C setting and then discretizing at the end. Is that correct?
Yeah, I mean, we're deriving, but ultimately the data we're working with does have finite, is voxelized. We have these image stacks from the camera. I mean, those effects are accounted for in the model in some way, that those voxels are much smaller than the spatial resolution set by the microscope itself, by the diffraction limit of the microscope. And the effects, the 2na over lambda limit, that is modeled in our H function.
Mm-hmm.
But it's definitely true that we would be sensitive to these effects, that if you had enough different, if you had, within a resolution element, if you had, sub-distributions of highly oriented structures, but that were all oriented differently from each other, I think those would average out over that volume and we wouldn't be sensitive to them. We'd get something more uniform. If we happen to have, like a bimodal distribution, like two crossing fibers below the resolution limit, you can't see the crossing fibers, but we might be able to pick up that fact that, okay, there's a lot of orientation in this data.
Yeah, no, that makes perfect sense. So basically, you need a good microscope that has good spatial resolution, and then you can, then you're in good shape. And then I just had another quick question, and this is just a general question. As you know, people are trying to learn priors for various inverse problems, and they're trying to fill in null functions. And so it's kind of refreshing that you weren't trying to do that. You were just trying to reconstruct the measurable component of the object and not move around. But I mean, do you think there is some opportunity for learning-based approaches to try to extend the resolution in some of these inverse problems?
Yeah, I think certainly for the same reason that it's worked in other domains. At this point, we're still, this is so new. We're still probing the sort of the basic physics and trying to understand what the what would even be reconstructible by classical methods and how to make that better. Like I think without the understanding we had, we would not have been able, had the idea of tilting the light sheet to improve the data quality, which I think would help any algorithm, even a learning-based one. But certainly I think there are priors to learn, especially if there are, patterns of orientation that emerge in certain, cellular structures, that, especially if there's sort of combinations of, spatial patterns and orientation, that could be co-learned.
Great, and maybe I just can't help one more question. Sorry, and let's say let's just assume you didn't want to do an algebraic type solution. So the solution you derived is very elegant, very beautiful. But what if someone just naively wanted to do an iterative approach where they had to compute the forward operator? And is that computationally feasible or friendly to basically compute the forward operator for a given distribution?
Yeah, it's tough. We thought about it a few times. The computational savings from coming into the spherical harmonic basis is so immense that we never saw any alternative to that. And I did spend some time, a lot of this work for me was done in the summers at the MBL, and spent, some time thinking about it. But my student convinced me that it was, sort of fruitless, given, taking this potentially immense sampling on the sphere down to just reconstructing at most 15 coefficients. So
yeah, it makes sense. Great. Well, thank you.
Hi, Patrick. I have a question. I'm a faculty in bioengineering.
hi.
Make molecular probes. This is a very exciting talk, and I was not familiar with all the developments that had taken place in an anisotropic and polarization imaging. Do you have any recommendations from somebody who makes probes and molecular labeling strategies to how to best maximize the development of probes to get the most out of an image, a polarization-based image from microscopy?
Yeah, it's a good question. We've had a few conversations with people at Genelia who sometimes come to MBL. I'm trying to remember what they were thinking. I mean, some of the keys are the I mean, obviously, very bright dyes are always useful for anything. Having sort of strong, well-oriented dipoles, and then the ability to connect with this highly rigid linker is key. Like, whatever, we're never interested in the dye itself, usually. I mean, there are some interesting problems where people use the dye, like the ability of the dye to rotate to report something about the viscosity of the environment that it's found in. And there are some classic fluorescence, bulk non-imaging fluorescence anisotropy methods that rely on that. But we've mostly been thinking in terms of imaging oriented structures. And so, yeah, having these linkers be able to be as rigid as possible is key, because if your dye is sort of flopping around, that's another, that's kind of a source of blur on your angular resolution. The other thing my student was thinking about was, there are folks who make dyes that are like sort of kind of fret-like dyes for force sensitivity measurements. And he was thinking about adding, trying to sense the not just the magnitude of a force, but its direction by making the right kind of molecular probe. And the details of what he'd talked to people about are beyond me, my limited chemistry knowledge, but yeah, that's another direction.
Is it always a bad thing if the excitation dipole is not identical in orientation to the emission dipole?
Oh, that's a good question. I don't think that's fatal. I'm trying to think. We talked about that at various points. I think most of our work has assumed they are parallel. But yeah, I don't think that would be a big problem. And it may even represent an opportunity in some ways.
Okay, interesting.
Yeah.
Thanks for the great talk.
Oh, sure, yeah. And then, yeah, certainly two photon stuff is also another potential direction, in that, there your excitation function gets squared. You need sort of two factors. And so then, these cosines, which are what give us angular resolution at all, everything goes up an order of magnitude, and that takes us down in the table of spherical harmonics, which is good.
So I saw a question from the audience in the chat. The question is, how do the polarization sensitive dyes maintain their orientation relative to the structure?
Yeah, that's sort of the key in the design of the molecule. So some of them just, I think some of the ones we've been using, like the phalloidin, the dyes that get added to cells, they're just known to sort of bind to, the phalloidin, which I think is a toxin in high quantities, will sort of bind to the actin molecules in a very particular orientation. It sort of locks on and is quite stable. For genetically expressed probes, like in the septin example I gave, there they engineered that yeast to express septin that had a GFP hanging off of it. There they had to take care to make sure that the linker, aminos between the septin protein and the GFP protein were likely to be very rigid, and short. If it's a long and floppy linker, then the GFP will just flop all around and it's not reporting anything. You won't even get an orientation signal.
Right. Makes sense. Cool. So if there's no more questions from the audience, I guess that concludes our first seminar today. So I'd like to thank Patrick so much again for giving us this wonderful talk.